So today I'm going to be talking about paracetamol, its synthesis, and a method to purity test it. So to begin with, what is paracetamol? Well, paracetamol, also scientifically known as acetaminophen, is a non-opioid analgesic and antipyrectic treatment agent used to reduce fever to mild and moderate pain, basically meaning that rather than binding to opioid receptors within the brain and spinal cord, it blocks chemical signals in the brain producing inflammation and pain. It is a widely available over-the-counter drug, commonly sold under the names Tylenol and paracetamol and Panadol. Unlike opioids, it does not produce an addictive effect or a high for the individual using it, making it a popular choice for pain relief. While its mechanism of action is not 100% understood, it has been theorized by scientists to inhibit the COX enzymes in the central nervous system, specifically COX2, leading to a reduced prostaglandin synthesis, which is the process in which the body produces pain and inflammation signals. This mechanism overall limits the pain and fever responses within the body without any significant anti-inflammatory effects. So now onto its history. It all began in 1878 when scientist Harmon Northrop Morse first synthesized paracetamol by reducing the p-nitrophenol with tin in glacial acetic acid. Acetanilide was also introduced to medical practice. However, the synthesized paracetamol from the p-nitrophenol was not widely recognized in medical practice due to its safety and ethical concerns. In 1893, Joseph von Mering and scientists in Germany investigated the derived and synthesized paracetamol's effects on humans. However, they noticed a slight tendency to produce methemoglobinemia, which is a rare blood disorder in which the hemoglobin does not produce oxygen signals towards the body, leading to its initial abandonment in favor of a similar drug, phenacetin. In the 1940s, Bernard Brodie and Julius Axelrod discovered that both acetanilide and phenacetin are metabolized to paracetamol, which is a better tolerated analgesic, and that it could be metabolized through these pain medications, making it more ethical compared to the first synthesized paracetamol from p-nitrophenol. Throughout the 1950s and 70s, after this discovery, paracetamol was first marketed in the United States under the name Tresigic, which also included caffeine and aspirin in its compound. It became available in the UK during 1956 and was sold under the name Panadol under prescription and was available for adults, children, and people with stomach ulcers. Ethical concerns delayed its safety until the 1970s, but in the 1980s, paracetamol sales exceeded those of aspirin in many other countries worldwide, making it very popular. Today, paracetamol is one of the world's most commonly used medications for pain relief and fever. It is considered a first-line treatment for many conditions due to its efficiency and safety profile when used as directed. Paracetamol has the molecular formula C8N9NO2, meaning that it consists of 8 carbon atoms, 9 hydrogen atoms, 1 nitrogen atom, and 2 oxygen atoms. Its physical structure also consists of a benzene ring, which is a 6-membered carbon ring alternating single and double bonds, known as a phenyl group, a hydroxyl group, which is a -OH attached to the benzene ring at the opposite side of the ring, and an amide group consisting of a nitrogen atom bonded to a carbonyl group, and a methyl group, which would typically look like this in the diagram below. Paracetamol is mainly considered safe when used correctly. Overdosing can cause significant problems, and one of which is liver damage. This occurs as excessive amounts overwhelm the liver's ability to metabolize the drug, leading to toxic byproducts. Individuals who use excessive amounts of alcohol and have pre-existing health issues should also remember to be cautious when using paracetamol, as it increases the likelihood of having toxic effects, with one being hepatotoxicity, which is also another term for liver damage. With regards to its safety profile, prolonged and excessive use can lead to significant health issues. Therefore, why healthcare professionals should advise responsible dosage recommendations and promote responsible usage. Now onto its synthesis. So paracetamol is synthesized primarily through the acetylation of 4-aminophenol with acetic anhydride, resulting in the formation of paracetamol and acetic acid as a byproduct. And paracetamol is typically synthesized in 3 steps, with these being nitration, reduction, and acetylation. So to begin with, through the process of nitration, the goal is to add a nitro group to a benzene ring in phenol, creating nitrophenol derivatives. The main things used throughout this process are phenol as the starting material, nitric acid as the substance that helps provide the nitro group, and sulfuric acid acting as a helper and catalyst, making the reaction smoother. Sulfuric acid helps the nitric acid produce a superreactive ion called the nitronium ion, and this is the thing that attaches to the benzene ring. The nitronium ion attacks the benzene ring in phenol, which has an OH group that makes the ring more likely to react, so the nitronium ion attacks the ring. The nitronium ion prefers to attach at the pair position because the OH group makes it easier for the reaction to happen there. Now, through reduction, the objective is to change the nitro group into an amide group, turning nitrophenol into aminophenol. To do this, we need to reduce the nitro group, which means adding hydrogen, and this can be done using a reducing agent such as tin or hydrochloric acid, or by using hydrogen gas with a catalyst. This process turns the 4-nitrinophenol into aminophenol, where the nitro group is replaced with an amide group. This reaction removes the oxygen atoms from the nitro group and replaces them with hydrogen atoms, forming an amide group. Finally, through acetylation, acetic anhydride is added to the reaction, and this chemical reaction contains an acetyl group, which is what we need to modify the molecule. The amide group in the molecule reacts with acetic anhydride, and when this happens, the amide group bonds with the acetyl group, forming a new bond called an amide group. And this process turns the amide group into turns the amine group into an amide group, and now the molecule in its final form as paracetamol. And this amide group helps make the paracetamol safe and effective as a medicine, more specifically in how it's absorbed and processed within the body. Now for purity testing. The purity testing method that I had chosen was thin layer chromatography, which is a method used to test the purity of a substance by separating it into its components, comparing it to a pure sample. The main materials for this process are a TLC plate that has silica gel or alumina, a solvent, either being hexane, ethyl acetate, a capillary tube used for spotting samples, a paracetamol sample, because that's what we're testing, a developing chamber, such as a small beaker or jar, a UV lamp for visualizing spots, and a pencil for marking the baseline, as well as a pipette for transferring substances. Thin layer chromatography is a simple and effective technique that's used to separate and identify compounds in a mixture. Its procedure goes like this. To begin with, you have to spot the TLC plate. Through this, you mark the TLC point with a pencil, typically 1 to 2 centimeters away from the starting point, and this is typically where you apply the paracetamol sample. You then dissolve the sample in a solvent like ethanol or acetone, which helps dissolve the paracetamol, and the solvent carries the compound up to the TLC plate, and then using a pipette, you transfer the tiny drop of this dissolved paracetamol solution onto the pencil line, making sure to apply it carefully so that the drop is small and doesn't overlap with other spots. Secondly, preparing the development plate. Through this process, you line the walls of the beaker with the plate, and you take a clean beaker and place the TLC plate inside so that it stands up, and the plate should not touch the bottom of the beaker. You then add the solvent to the beaker, such as hexane or ethyl acetate, and this mixture helps the paracetamol sample separate on the TLC plate. Finally, you'll cover the chamber and keep a lid on the beaker to prevent solvent vapors from escaping and to prevent contamination. Thirdly, through developing the TLC plate, you place the TLC plate in a chamber and into the solvent-filled beaker, and this solvent should rise through the plate, carrying the paracetamol sample within it. As the solvent moves up to the plate, it'll carry the paracetamol sample at different rates depending on the polarity of the substances within the mixture. Paracetamol will then form a spot at the specific point, and then remove the plate when it's close to the top. Through the fourth step, when visualizing the spots, Although the spots cannot be visible towards the naked eye, you'll use a UV lamp to identify them, use a pencil to circle the spots to determine where the impurities are, and then turn off the UV lamp when done. Finally, you calculate the RF value for each spot, which is the spot distance over the solvent distance, which gets divided by each other. And then through this, you are able to determine the impurities, how many there are, and the extent to which where they are. Overall, paracetamol is a widely used pain reliever with a well-established history, clear medical benefits, and very important safety considerations. Its synthesis follows a three-step process, with these being nitration, reduction, and acetylation, making sure that it has an effective final product. To confirm its purity, methods like thin layer chromatography provide a simple and reliable method for identifying its impurities. While paracetamol remains to be an essential form of medication, responsible use and an awareness of its risks are important to know. Understanding the medication as well as its chemical makeup not only highlight its medical significance, but also illustrates the importance of quality control within pharmaceuticals and medical settings. Thank you.